Hello, my name is Jason Linville. This is the lecture on forensic DNA analysis, part one of two parts for JS110 Introduction to Forensic Sciences. This lecture is being recorded in June 20th, Camp Week of 2017. So if you're accessing this archive lecture in future semesters, anything I say about the summer of 2017 doesn't apply to you, but all the material in the lecture still applies. One of my other students was harassing me about introducing myself in the beginning of all these lectures. And I say that's just in case it's used in another class or if it's used in another semester, just so everybody's clear about when this is recording and what's going on here. For the summer of 2017, where we're at is I screwed up the lecture last week. So I think so Garrison's in the room this week, even though a lot of people from last week got credit for it. But just sort of as a reminder, summer 2017, look at the schedule. I'm going to try to add some dates to the calendar and jump in one of these live lectures. For this week, again, summer 2017, you have your biological fluids, other body fluids. I forget the current name of it, but we talk about semen, saliva, and some other stuff. And then also the DNA lecture is in two parts. The reason it's divided up into two parts is because if we went over everything, it would be pretty long and awful, especially for an online lecture. But this way, part one sort of covers some background of DNA. So if you've recently had a lot of biology classes, then this may be some review and boring. But if you haven't had biology since high school, then this is sort of a refresher until we start talking about what goes on in a forensic DNA lab, which we'll pick up with the next lecture. All right, so let's go ahead and begin with forensic DNA analysis part one, if I can get to the screen where I'm actually changing the slides. Okay, so we're going to go over in this lecture short, what is DNA, where is it found in the body, how is it different among individuals, because most of us probably understand, hey, you find DNA at a crime scene and you compare it to the suspect and you can say that blood came from that individual. So in theory, DNA must be different among all individuals. And then in part two is where we'll get into those last two bullets, the forensic DNA analysis and the DNA and statistics. Okay, number one. I don't know why I said number one. It's been did I mention it's camp week? Long day. All right, one of the first things we have to ask ourselves when we're talking about what does DNA stand for? Okay, well, lucky for you, I have a little story I like to tell about one of my good friends who I don't know. Her name is Nancy Grace. She has a television show on some network, and she likes to accuse people that she thinks did something wrong of definitely doing something wrong. And back during the, what was it? It was the Casey Anthony, Caylee Anthony case. And don't worry, I'm getting to DNA here. And Nancy Grace, was convinced that the mother had done something to her child. So what's she going to do about it? Well, she's going to go out and she's going to find a guy with the biggest cowboy hat she can find, and she's going to bring him onto her show and ask him questions. And apparently this guy knew like some cops or some of the investigators, and he was like breaking news. And one of the things he said, well, Nancy, they found some DNA in the trunk of the car. And Nancy Grace is not just a go-get-em journalist / TV personality, but she also likes to educate her audience. So her response to this statement that they found DNA in the trunk of the car was, and if you're watching on the webcam, this adds something to it. If you're on audio, I'm holding up my finger and pointing to the sky and sort of shaking it in unison here. She said, whoa, whoa, whoa. Are you telling me that DNA, deoxyribonucleic acid, was found in the trunk of that car? And what she did there was she defined what DNA was and sort of threw in a little education to let you know that she knew what DNA stood for. And now you know what you know, which is DNA stands for deoxyribonucleic acid. Now, don't be confused. It can be written a couple different ways as we see here on the slide. So on the left, deoxyribose nucleic acid, it's real clear where the DNA comes from. But don't get confused, because if multiple choice question, what does DNA stand for? It may be written out as deoxyribonucleic acid, and the N of the DNA is sort of hidden in there. All right, what does that mean? It's a nucleic acid as a deoxyribose sugar, but that's beyond this class. We're not going to worry about it. All right, what does DNA do? Well, Most of us understand, well, DNA does, if there's a genetic mutation, if there's something wrong with your DNA, then you either have a disease or supernatural powers. Um, but it contains genetic information, and we're going to try to learn more about exactly what that means in this short lecture. The genetic information that it contains basically means that it codes for making proteins, as it says there, that are necessary for survival. If you remember in any high school biology classes, you may have talked about, transcription, where DNA codes the messenger RNA, then it goes to the ribosome and it makes a protein. All right, the way I want you guys to think about these proteins is they're sort of like little nugget machines that your body can make. And there's several different types of protein that the body can make, which we'll go over a couple of them. But maybe this, maybe this helps you recall some things. DNA is a series of letters, A, G, C's, and T's, and we'll review what those are in a couple of slides. But it's read in series of 3, in triplets of 3, and each of those 3 letters will code for a different amino acid. All right, so it's sort of like having a recipe book almost, okay? So your body reads the DNA and sees a TAG and it's say, okay, that is a leucine or some type of amino acid, and then it adds another amino acid to that, and this chain of amino acids folds up and forms a protein that does something in your body. All right, what is something? Well, we already learned about one protein your body makes, and that's hemoglobin. Okay, chain of amino acids folds up and then it floats around in your bloodstream, binds oxygen in the lungs, deposits it to the cells that need oxygen. Okay, insulin, when you eat, your body makes insulin. It turns on this protein that your body makes. Your body needs to break down food. It needs to turn starches into glucose and glucose into energy by breaking it down, and it does so with enzymes. Enzymes are proteins that your body makes, okay, and are present in your body. During early development, let's take males for example, on the Y chromosome is something called an SRY gene. It makes a protein that then goes to other areas of DNA and turns on other genes, and so it's starting to make all these proteins that causes an embryo to become male as opposed to female. All right, so not just the proteins that you make, but some proteins regulate how other proteins are made. So because our DNA is a little bit different, it affects the way these proteins are made, and it is what makes everybody a little bit different from somebody else. All right. So these proteins are constantly being made and used and broken down by your body. All right. But since we all look a little bit different, then we know that all of our DNA is a little bit different, and that leads to these proteins being a little bit different. All right. If you think about it, really the only 2 people that look exactly the same are identical twins, and we're going to get in it. Identical twins have the same DNA, so they're making the same amount, relatively so, the same type of proteins. They have the same mutations, and that leads them to look the same. All right. Okay, so the fact that we all look different implies that our DNA is different, because our DNA is what controls what our body makes, how our body grows, and what our body looks like. All right. What does DNA look like? Well, you may have heard these terms: double helix, twisted ladder. Here's a couple different diagrams. Whoops, this is terrible. I've changed the animation, so now everything's going crazy. DNA is a double helix, which we can see if you untwisted that ribbon on the left there, you would see it looks like a ladder, okay, where we have sides of the ladder and then the rungs of the ladder. This is a model, okay? If you get a really powerful microscope, you still can't really zoom in on DNA and see it, so that's not how we're going to tell what order the A, G, C's, and T's are. We're going to measure it indirectly some other way. But this is sort of a model of how we want to visualize DNA, okay? So there's several different ways DNA can be modeled, several different ways DNA can be drawn, okay? But no matter how you draw it, whether it's a cartoon drawing on the left or whether it's a model of where all the molecules and atoms we think are on the right there, keep in mind that it always has the same format where it's this twisted ladder. So what we have there, the sides of the ladder, if it was untwisted, are what's called the sugar phosphate. Okay, so it means it's a sugar group and a phosphate group. But what's important for you to know is that the sugar phosphate in every single person, okay, if we were to compare my DNA to Garrison's DNA, we have the exact same sugar phosphate sides. Our sides of the ladder look exactly the same throughout all of our DNA. I guess you could say we're identical sugar phosphate twins. Don't say that. Okay. The rungs of the ladder contain the nitrogenous bases, which, as we're going to see, are the, if you look at that cartoon on the left there, those are the A, G, C's, and T's. And those A, G, C's, and T's are not identical if we compared Garrison's DNA to my DNA. So it's really the rungs of the ladder that are going to be different among different individuals. Okay. How is a ladder constructed? Well, if you were going to build a ladder to get on your roof or whatever, go buy a ladder. Don't build your own ladder. That sounds dangerous. But if you insisted, you would probably put like one rung on top of the other, right? Sort of have a couple of H's and stack them on top of one. Okay, but how I want you to visualize DNA, and if you were building DNA, is you're not going to put one rung on top of another. You're going to put half of a rung on top of another. So if we look there, and let me think, because I think in this one, I'm recording lectures like differently here, but I think in this one I can actually draw on here. There. Are you seeing that, Garrison? The drawing on there? Okay. You can see this looks like a sugar phosphate. This is the phosphate here, the P. Okay. This is the sugar here, the S. That's the side of the ladder. Okay. So we have our sugar phosphate here, P and S. And then the rung of the ladder, it's a half of a rung of a ladder, and that's a base. Okay, and that can be an A, C, G, T. Because I look at this stuff all the time, I'm pretty sure that's an A here. Okay, so how this ladder is built is it's with like half rungs of ladder on top of one another. Okay, so it's phosphate, sugar, phosphate, sugar, phosphate, sugar there. Okay, and then we have like an A, a G, a C, or a T, or whatever. Okay. And then the other half of the ladder is constructed by your body to complement the ladder that's already there. Okay. So don't just think of it as a rung on top of another rung. Think of it as two half ladders that are paired together. Okay, this is going to make more sense later on. But the building block is a nucleotide, half of a rung on the ladder. Okay, so that half of a rung of a ladder can be an A, G, C, or T. And here, if we look at the molecular structure, oh, Dr. Limbaugh, I've never taken chemistry. I don't even know what that N is. Why? Okay, don't worry about the molecular structure. Okay, I want you to think of it as the side of the ladder that is a sugar phosphate, and think of, okay, understand there are four bases, an A, G, C, or T. So you can picture it like a model with the letters. Don't worry about the molecular structures. Okay, so this is how I want you to visualize DNA. We have the side of the ladder, okay, the sugar phosphate side, and you don't even need to worry too much about the P or the S's, and that O is an oxygen, so ignore all that. Focus on the middle part, okay? So notice how half of the ladder here is a TTGC, all right? And then the other half is constructed, and your body just knows how to match it up because it can't, on the other side of a T, A is really the only other base that will fit in there, okay? So A complements T. So wherever you have a T over here, you're going to have an A on the opposite side. Wherever you have a G over here on the left, you're going to have a C over here on the left. And as you can see down here, if you have a C on the left, you're going to have a G on the right. Okay, excuse me, I think I said it incorrectly. If you were to have an A on the left over here, which we don't see, then you would have a T. A always goes with T. G always pairs with C. Okay, so in other words, this is the take-home message. If you know the sequence of letters on one side of the ladder, you know the sequence on the other side. So if you're writing the sequence of letters on one side of the ladder, you don't have to write the sequence on the other side because you already know what they are. Okay? So all you really need to do if you're talking about DNA or describing DNA, whether it's in a advanced scientific paper where you're curing cancer, or if it's in court where you're describing DNA to a jury, all you need to do is talk about that one side of the ladder. Okay? So this is how it's written. Notice that this is not written as A paired with T or A, double pair with T or A dash T or anything. No, we write A. We're writing the sequence of DNA. We're writing one side of the ladder. We know the other side is T, T, C, A, but we don't need to write it, okay? We don't have those keys on our typewriter anyway. So if we're looking at a piece of human DNA, we can describe it by writing out the letters in sequence, and we just have to write one side of the ladder, okay? All right. In humans, if I were to ask you how many base pairs, how many nitrogenous bases are there, okay, the answer would be 3 billion. Okay, this is incorrect. And I always talk about how it's incorrect or a little bit wrong. I should just go back and change it, shouldn't? Hey, maybe version 2.1. Okay, in humans, there are 3 billion base pairs in one copy of the DNA. Okay, in one copy of the DNA within each cell. Now that's going to become important later on because we're going to realize that because you have a mother and a father, you actually have 2 copies of DNA in each cell. Okay, but if we were just talking about human DNA and what the sequence of DNA was, we could talk about either one of those copies, and that would be a sequence of 3 billion. I'm trying to get you to understand how many of these bases are in one copy of the DNA. Okay, so you get 3 billion letters from your mother, and you get 3 billion letters from your father. If we're talking about human DNA, we just talk about these are the 3 billion letters for human DNA, and this is the order of those letters. Okay? Oh, I went too fast. That's okay. Garrison wasn't paying attention, right? All right, so I want Garrison in the chat box, if he's paying attention, or you at home, to give me a percentage of if we were to compare Garrison's DNA and my DNA, okay, how similar do you think our DNA would be? Okay, we're both humans, I'm assuming. Okay, so are Garrison and I, are we going to be 10% similar, 25% similar, 50% similar, 90% similar? All right, Garrison, okay, so take your time, pause the lecture because I'm going to reveal the answer. Take your guess, throw out a number. Garrison says 95. That's a good guess. Okay, it's a little bit higher than that. For humans, it's 99.8% similar. Okay, so if we were to write out, if Garrison and I were to write out all 3 billion in one copy of our DNA and compare it, we would find that we're 99.8%. That's very similar. And a lot of people, I mean, we as humans, we focus on looking at each other and seeing all the differences. Okay, but if you think about it, Garrison probably has 10 fingers, 2 eyes, a circulatory system, a respiratory system, a functioning brain, teeth. Okay, if Garrison eats food, he probably breaks down glucose and uses it for energy. His cells probably have proteins to do oxidative phosphorylation. Am I on the money here, Garrison? Am I like describing you to a T? Okay. All right, because that's what all humans are like. In fact, if we even go to, as it says there, chimpanzees, who also have hair, circulatory system, 2 eyes, lungs, eat similar type food, etc., etc., 95% of the things that makes us animals and mammals, okay, are very, very similar. Okay, so you can even go down the line. Your DNA is still pretty similar to a dog's DNA because, again, we're both animals, right? Okay, so 99.8%, that seems very high, but it's only that small 0.2% that still leads to all of us looking quite different, having different disease, growing to different sizes and shapes. Okay, so a lot of things that we can see. But, if you want to put a number on it, all of us humans are pretty similar. All right. So now, as forensic scientists, because if you're researching like genetic diseases, you focus on the parts that are the same, and then you look at people who are different from what is, normal, let's say. Okay. Or people who you look at the people who have that genetic disease versus the people that do not, and you sort of find to see what those unique differences are. But for forensic science purposes, we don't care about the weird mutations. We don't care about the areas that are the same among all the individuals. We will care about those areas of DNA that are different and don't really cause much of an effect. Okay. There are locations in our DNA where Garrison and I we know we're going to be different, and we know all humans are going to be a little bit different in these areas of DNA. So that's the area that forensic scientists focus on. Not the 99.8% that's going to be the same. We focus on the 0.2% that's different. I title this slide Stupid Facts because they are facts, and they are stupid, although that's a matter of opinion, I guess. I think these mentioned in the book, it just sort of tries to emphasize the similarity of two different individuals' DNA. So if Garrison and I are at a party, and we decide that we would start reciting our DNA, you know, to pass the time, and maybe to impress other people at the party. Okay, so we stare each other in the eyes and start going A, G, C, T, you know, and we're the same. We're 99.8%. It would be about 8.5 minutes before we'd get a letter that's different. Well, that's a dumb example. All right, if unwound and tied together, I like where this is going. Your strands of DNA would stretch about 6 feet tall in one cell, but only 50. So there's a lot of information crammed into a tiny, tiny cell. In fact, if you took all of your DNA and put it end to end, you could go to the sun and back 600 times. If you don't believe me, look at the math there. 100 trillion cells in your body, 6 feet of DNA in each cell, 92 million miles to the sun. All right, let's get out of this slide. Okay, so that's what DNA is. It's a series of letters connected together. Where is it found? It's found in the nucleus. That's the DNA that we're going to talk about. We're going to talk about mitochondrial DNA a little bit later, but nuclear DNA is found in the nucleus, okay? If you find blood at a crime scene, you're going to analyze the nuclear DNA. It's only in certain circumstances that we'll look at the mitochondrial DNA. All right. So we're going to focus on the nucleus there. We'll come back to mitochondria later. Okay? In the nucleus, these are some cells that have nucleuses. Nuclei? I don't know. Okay, white blood cells, red blood cells lose their nucleus, but in your blood, even in a tiny drop of blood, we already went over this with blood ID, you have plenty of white blood cells that have DNA in them. Sperm cells have DNA. If you're advanced in biology, you may say, wait a minute, Dr. Linville, isn't sperm cells haploid and they only have one copy as opposed to... Yeah, but if you take 1,000,000 sperm cells, or let's say 1,000 sperm cells, it has the same DNA as 500 blood cells. So in a biological stain, a blood stain has the same DNA as a semen stain. Cheek cells also have the same DNA is in all of these cells. So that's why if we have a semen stain that we're using as evidence, we don't have to get a semen sample from our suspect to compare the two as far as DNA goes. All we need to do is get DNA from that person. So we can swab a person's mouth and the DNA from a person's cheek cells is going to be the same profile as the DNA from the sperm cells. Well, why do the cells look so different? My short example of why this is the case is imagine that every cell has the same Betty Crocker cookbook. Okay, the DNA are the instructions on how to make these proteins, right? So every cell has the same Betty Crocker cookbook, but each cell chooses to read a different page of that cookbook and express different proteins. Okay, so blood cells are reading page 74 and they're making pancakes. Cheek cells are reading page 76 and they're making blueberry muffins. I guess we really haven't gotten out of the bread family here, but you get the general idea. Same DNA in each of those cells, but it's just different parts are being read and expressed, and that's why cells look different. A brain cell looks different from a cheek cell, looks different from a liver cell, looks different from a blood cell. But if we scoop them up and analyze them in a DNA lab, they have the same DNA in them. Okay. How do you get 6 feet of DNA wound up into a tiny cell? It's twisted and wrapped around histone proteins, and it's really crammed in there. Okay. So it's crammed into things called chromosomes, which is basically just a short strand of twisted up DNA. Okay. Now, you can't get all 3 billion letters twisted up into one big chromosome. So our chromosomes are constructed to where there are 23 of them. All right. And in each cell, we have 23 pairs of chromosomes. So let's go over these numbers because there's 46 there, there's 23. I've been saying 3 billion. Okay. Let's reset our brain here. Okay. You'll notice that you have 23 pairs of chromosomes, so you have 2 chromosome number 1s. Okay. One of those chromosomes you got from your father. Little male symbol there. One of those chromosomes you got from your mother. Okay. Chromosome number 2. One of those you got from your father. One of those you got from your mother. Okay. All of the chromosomes that your father gave you, 23 of them. Okay. Oh, XY down here, so we know the father gave the Y. All 23 of those chromosomes, if you added up the letters, the A, G, C's, T's, and again, if you zoomed in on any one of those chromosomes, you would eventually get to where you see those A, G, C, T's, the twisted ladder. If you added up all those letters, you would get 3 billion. Okay? Your father gave you 3 billion base pairs. Your mother gave you 3 billion base pairs. Okay, that's the other chromosome set. If you added up all of these 23 chromosomes that your mother gave you, it's 3 billion. Okay, so that's why you have 2 copies of the human genome in each cell. And if you compare those 2 copies, the copy that your father gave you is 99.8% similar to the copy that your mother gave you. Okay, and if you take it as a whole, you know, it works out to where you're 99 or 0.8% similar to another person if you compare both of your copies of DNA. Okay, so that can get a little bit confusing, but that's the way it is. All right, this is going to become important in the next lecture when we start to talk about alleles and loci and stuff like that. Okay, all right. So where does DNA come from? I mean, we talked about it comes from a biological sample. We know it comes from semen. We know it comes from blood. It comes from anything that has cells in it because remember, the DNA is found in the nucleus. The nucleus is found in cells. Now, the reason this is important is because, and I don't really ask too many questions about this in this class. This gets a little bit more advanced. JS 352 offered this fall. But for blood, semen, and saliva, all of those are liquids where the cells are just floating around in them. So if you have a blood stain, you know, you just cut it out and try to get DNA from the blood cells floating around. If you have semen, you cut out the stain, you try to get DNA from the cells that are floating around in it. Saliva stain, you cut it out and you try to get DNA from the cells that are floating around it. Okay, but then we get into tissue. If you have a chunk of liver that for some reason you're using as your forensic science sample, I don't know, it's a drug ring where somebody's woke up in the bathtub and their liver is missing. I don't know. Okay. But liver is like liver cell stacked on liver cell, and it's like a whole bunch of cells. So you take any small chunk of liver and you're going to get a lot of DNA because it's composed of a lot of cells. All right. Now bone, this is where we start to get a little bit different. Bone, the exterior, the hard white part of bone actually isn't made up of cells, but it's like an extracellular hard structural material. But the bone marrow contains living cells that help produce, you know, blood in your body. Okay, so if you find an old bone in the woods that you're trying to get DNA out of for identification purposes, you're not just going to scrape a chip off of the outside white part of the bone. You're going to focus on drilling into the bone and sampling that bone marrow that is in the center. You're going to have much more success. Okay, teeth, same thing. The enamel, the hard white part, no DNA. But the inside of a tooth with the pulp, that has DNA in it. So you could sample hair. I think we may have mentioned, or no, wait, we haven't had trace yet, so we haven't mentioned this. Hair, the end part of hair doesn't have much DNA in it. It has a little bit. We'll get to that. But if hair is ripped out of a person's head, you know, in the process of murdering another person, then the roots that come out at the base of the hair, those do have living cells in it. Okay? So if you're a forensic scientist and you get a hair sample, you want to look at the root and sample that for your DNA. So it's all about understanding the sample that you receive and knowing where the cells are in that sample. And that's what you target for your DNA analysis, because that's where the DNA is. We'll ignore maggot crops. That has to do with my dissertation research. That's not important right now. Okay. How does human DNA differ among, or how does DNA differ among humans? We already said that it's 0.2%, but out of 3 billion, that's plenty of DNA that's going to be different between Garrison and me. Okay, we're going to have no problems finding areas of DNA where if there's blood at a crime scene and the question is whether it came from Garrison or whether it came from Dr. Linville, we'll do that analysis and we'll get an easy answer. Okay. DNA is a sequence of 4 possible letters, A, G, C's, T's. And remember, we're just talking about one side of the ladder here. One side of the ladder is 3 billion letters long. Okay, 99.8% between people is identical, but let's focus on how can it be different. Okay, there's 3 different ways. The first one is probably the most common. If you say, well, how could DNA be different between individuals? You say, well, the letters are different. We'll look at some examples here. Also, letters can be added or moved, and it's really that last one that's the most confusing one, unfortunately, that is what forensic scientists look at. Okay, so let's say we're looking at your DNA and we're looking at my DNA. Okay, we can see there that, oh, okay, it's very similar because we're both human, but right there, we have a base that's different. Okay. Now, notice here I'm sort of cheating because I'm just looking at one copy of your DNA and one copy of my DNA. All right, so let's just ignore the fact that there's 2 copies and you may actually have a T and a C, or a T from your father and a T from your mother. So let's just focus on how can DNA be different, okay? You could have a T and I could have a C. All right. But the problem is, there's only 4 options there. You can have either a T or a C or an A or a G. When it comes to differentiating the billions of humans on Earth, okay, that's not that strong, okay? Because my DNA is not going to be unique. Somebody else is also going to have a C. And for you, somebody else is also going to have a T. Base pairs can be added or removed. So here we can see that you, me, and him all have the same sequence, and then right after the G, you have 2 T's, I have 1 T, and him has 3 T's. Okay. There can be additions or subtractions in there. Okay. If we look at her, we'll notice that a big chunk of DNA is missing, so she's a little bit different. And this is possible to, occur between people. And it's possible for forensic scientists to use this to analyze this area of DNA and find out, who the blood matches. If the blood has 2 T's, then it came from you. If it has 1 T from me, 3 T's from him, and if their big chunk is missing from her. All right, but that also turns out to be difficult to do that type of analysis. Okay. What we do in forensic science is we look for areas in your body where DNA is repeated, is repetitive. All right, can somebody see the pattern here in this DNA? Feel like I'm on an episode of Team Umizoomi here. All right. Here, let me change the colors for you. Boom. You see that? You, me, and him, we all have the same sequence of DNA up to this point in our DNA. And then we all have AGCT. But the difference is you have AGCT repeated 6 times. I have AGCT repeated 5 times. And that other guy has AGCT repeated 7 times. Okay. So if we were to analyze this area of DNA in a blood stain at a crime scene and measure the DNA, measure the number of repeats, and it came back as having 5 repeats, we could say, oh, well, it didn't come from you and it didn't come from him. All right. So it had to come from me, the me in there, or somebody else with 5 repeats. Now what we're going to talk about in the next lecture is we're going to talk about, okay, how do we actually go from analyzing this repeated DNA to getting a DNA profile from a blood stain found at a crime scene that we can go into court and say this blood profile matches Dr. Linville's DNA, and I know no other human on Earth can have that same DNA unless he has an identical twin, which I don't, hopefully. All right. Okay. All right, so that's it. This DNA part 1 is, I think, one of the shorter ones. So Garrison got off the hook getting his credit here. Garrison, I do have you down for attendance, so you can, at this point, drop out at any time. I will hang around. Garrison, if you do have any questions about lecture, throw it in the chat box, and I'll answer it so the rest of the class can hear. For anybody else watching this lecture in whatever semester it may be, if you have any questions about the lecture, send me an email, drop by my office, and I'll try to give you an answer. For summer of 2017, go ahead and watch DNA Part 2 while it's fresh in your mind, because we're going to go more into depth to this slide right here. And then I believe you have another quiz at the end of this week, the mock case quiz, which sort of read about those mock case quizzes, try to prepare for it, and you should be in good shape. Okay, good. Garrison asks a great question. What would be an example of a case question? All right, Garrison, I'm going to give you an example of one for blood identification, which the lecture we had last week, because pretty much this mock case quiz is going to cover everything from the beginning of the semester to right now. Okay, so an example, these mock case questions, I describe the analysis and then you have to come to the correct conclusion. All right. So I may say you have a suspected blood stain that comes into your lab, okay, a red stain. You perform a TMB test on that stain, and the stain turns blue. What is your conclusion about that stain? The multiple choice answers are, and Garrison, you can take a shot at this, and you at home can guess as well. A, it is definitely blood. B, it is definitely not blood, or C, it might be blood. Of those three options, if you test a red stain and it turns blue for TMB, is it definitely blood? Is it definitely not blood? Or is it maybe blood? All right. Now, this is something where if you haven't watched the lecture, it may be difficult to understand because you need to understand, well, wait a minute. Is that a confirmatory test? Is TMB a confirmatory test where only blood is going to turn blue? Or are there other things that are also going to turn blue? All right, Garrison has it right. Hopefully you have it right where it's maybe, because that's a presumptive test. And we went over chemical oxidants may turn it blue or plant peroxidases. So it doesn't mean it's definitely blood. Now, in the actual case question, there's probably going to be two maybe answers. Okay? Number one will say, maybe it's blood because other things may also turn blue. That's the correct answer. But then I'll give you sort of a red herring. I'll say, maybe it's blood because the presence of hemoglobin can change over time. Well, yeah, hemoglobin can disappear over time, but that's not why in this situation where it did turn blue, that's not the correct maybe answer. Okay? If it didn't turn blue, then that would be the maybe answer. But here, so in other words, there may be two maybe answers, and you have to apply your logic to figure out which one is the correct for a given situation. All right. With that being said, if you've already watched the other body fluids lecture, there may be a question like, you receive a semen sample. It tests positive for the AP spot test. You perform a Christmas tree stain test on that stain, and you see sperm cells. What is your conclusion? A, it's definitely semen. B, it's definitely not semen. Or C, it's maybe semen. All right, if you don't study for these, you want to answer maybe to all of these answers. But what would the answer be to that question? A Christmas tree stain test where you see a sperm cell. That's a confirmatory test, right? So the answer to that would be A, it's definitely semen. This is not high school true and false question where if you see the word always, it's all, that means it's false, okay? Sometimes definitely yes is the answer. Sometimes definitely no is the answer. So you need to be familiar with these tests, understand what conclusions you can draw from them, and that's why it's kind of hard to look up the answers to these. You can use your notes to help you out, but you really have to watch the lectures and understand, and really start to understand the difference between the presumptive tests and confirmatory tests. All right. So good. I'll try to post this. The quiz is multiple choice. So yeah, your multiple choice answers, Garrison, will be like A, definitely yes. B, definitely no. C, maybe because blah, blah, blah. D, maybe because blah, blah, blah. Yeah. And hey, yeah, it's a good deal for me too, because, grading them, multiple choice is easy grading, right? All right. So hopefully, you take this first one as long as you prepare for it a little bit. If you've watched this posted DNA lecture all the way to the end, then you get some, bonus help information. Feel free to spread that around to your friends to watch the end of the DNA Part 1 lecture. All right. And hopefully everybody does good on this first mock quiz coming up on Monday. Excuse me. All right, that's it. Garrison, if you have any other questions, I'll hang around for a little bit. You can still ask me in the chat box after the recording stops. For everybody else, send me an email or stop by my office this summer so I get to see you in person, and I'll try to help you out. All right, good luck.